this pandemic situation will clear in that time. I promise I will give you the every type of that.
Don't say if.
Didi, how are you running the question answer session?
Sir, actually, the question answer session will be written in the chat box, sir. Written in the chat box. And if anyone want to unmute, then they can voice call, sir. No problem. If 2 to 3 at a time open, then there is some problem. They have to write in the chat box. If one by one, then they can speak. Unmute.
Yeah, I think Titi, the chat box use would be better because
yes,
otherwise, sir. Otherwise there will be many questions at the same time. Then it will be a problem.
Yes, sir. Yes, sir.
Yeah,
yes,
only Professor Pine can answer all this.
Yeah, I can see. I can.
If you open all the voices, there will be problem.
Right. Let her also listen what is going on. Huh? Devasisda, now I know where did I get this information from. That day I was telling you, I did not.
About?
What did I came to know about?
No, from his banner, from her banner.
From the banner, yes. Now I know.
I could realize that time immediately because I posted it in Facebook also.
Right, but that time I couldn't remember. I was thinking that maybe the PhD students, they wrote as a letter of recommendation, they put the list of references in their applications. I was thinking about that, and then I lately realized, no, it is not that.
আমি জানি আমি কি করব বলো আমি ঠিক আছে আচ্ছা কিছুই আর হ্যালো
Yes, we can hear you.
Okay, sir.
So, Titi will start exactly at 2:30.
Yes,
many people are joining.
Yeah, okay, sir.
Right.
Sir. চলে যান নিয়ে খেলে চলে যান হাই রে কথাটা
Sayanti joining from Germany.
Good. Teacher's lecture.
Sir, Devasis Rai, sir?
Yes.
Yes, sir. Sir, you can start, sir.
I can start?
Yes, okay. Sir.
So, good afternoon, everybody. So, the next speaker of this session will be Professor T.K. Pine from Indian Association for the Cultivation of Science. And truly speaking, he is one of my beloved students. But I produced so far during my 29 plus years of teaching in IIT Kharagpur. So he did his MSc in 1998 and he carried out his graduate studies in Max Planck Institute for Bioinorganic Chemistry in Germany. And after getting his PhD degree in 2003, he moved to University of Minnesota, USA to carry out his postdoctoral work. And after another stint of 3 years, he came back to country. In 2006, he joined ICS and currently he is a professor of that chemical science group of ICS. And he is a very prolific worker and particularly partly he's doing synthetic protein chemistry and catalysis, and he's publishing a lot. And his area of research includes particularly the biomimetic inorganic chemistry and bio-inspired catalysis, as we know, because we get the ultimate inspiration from nature. And how nature, particularly he is a master in iron chemistry, iron biomimetic chemistry. So how we can understand all these things, because from our college days or school days about iron in bioinorganic chemistry, we know about myoglobin and hemoglobin. But beyond that, what other things are there related to the iron biochemistry or iron bioinorganic chemistry? And during the last, say, 14 years, he is doing all these things in his laboratory. And mechanistic studies, he is also studying on O2-dependent oxidations and other transformations. Okay, so with these few words, I request Professor Pine to deliver his talk. Professor Pine, please. Taman, you start, Taman.
Yeah. I'm just trying to share my slide now. Okay. Yeah. Okay. Thank you, sir. Thank you very much for the introduction, and I'm grateful to Tiffy for having me here in this very unusual presentation, because all of we are going through a very unusual time. So we all have to be used to with this unusual mode of lecture, particularly when you don't see the face of the people in the audience. Anyway, let me get into that lecture today. What I'll be talking to you is that our efforts towards understanding, as Professor Ray mentioned, that one of the major objectives in our lab is to understand how Mother Nature uses oxygen, particularly for aerobic organisms, to perform a versatile array of oxidation reactions, and trying to understand the enzymatic reactions using simple synthetic compounds. And here, as Professor Ray mentioned, that synthesis is very, very important. So making molecule is the clue. And here I'll be showing you how we got the inspiration from nature and how we progressed. I will show you only one example today, particularly for one enzymes, how we understand over the last almost 7 to 8 years and where we stand now. And this enzyme is nothing but it's an aliphatic CC bond cleaving enzymes. And we have performed a number of mechanistic studies, and we came to a point where now we understand a little bit about that enzyme. But before we get into that enzyme, let us see how Mother Nature uses oxygen for performing versatile oxidation reactions. Some representative examples are shown here, that the iron-containing enzymes that activate oxygen from its triplet state, because its triplet state is not reactive. Examples include alpha-keto acid-dependent oxygenases that perform a CH bond hydroxylation reaction with the incorporation of oxygen atom from O2, and that use an alpha-keto acid, particularly alpha-keto glutarate, in the enzymatic system to carry out this reaction. And the cofactor alpha-keto acid provides electron that is necessary for the reduction of oxygen in this reaction. And in the reaction, one oxygen atom from O2 is incorporated into this aliphatic CH bond to form the hydroxylated product and concomitant with the decarboxylation of alpha-keto acid. Another enzyme, which is a bacterial enzyme, risky dioxygenases, as you can see here. Let me find out where is that. Just a second. Let me check where the. Here I got the laser pointer. Yeah. So here you see that the risky dioxygenases It's a bacterial enzyme which is involved in the degradation of toxic pollutants, particularly polyaromatic hydrocarbons. And in the first step of the degradation, the enzyme catalyzes the cis-dihydroxylation reaction. Very unusual reaction you see, where both the oxygen atoms are incorporated into the aromatic ring, forming a dihydroxylated product. And once this functionalization takes place, subsequent CC bond cleavage is relatively easy by some other enzymatic systems. Similarly, in case of an heme enzyme, most well-studied cytochrome P450, which is involved in the aliphatic CH bond hydroxylation as well as epoxidation reactions. So in all these enzymes, these are oxygenases. That means oxygen atoms are incorporated into the product, and all these reactions take place under ambient condition using oxygen as the terminal oxidant. Terminal oxidant means it actually accepts electron, and eventually the reduced oxygen species performs the oxidation reactions. It's not the O2 which directly involved in the reaction. So how they normally, the accumulated mechanistic information that is now known indicates that in all these enzymes, in the resting state, when enzyme doesn't do anything, iron 2 plus is present in the active site. And at the iron 2, with its unpaired electron, interacts with the oxygen molecule, particularly the anti-bonding orbitals where the electrons are there. Then it forms initially an iron 3 superoxo species, and this superoxide can either accept an electron, or in the presence of a cofactor, it can form an iron 3 hydroperoxo species or an iron 3 alkyl peroxo species. And then in the OO bond cleavage of the oxygen-oxygen bond of this alkyl or hydroperoxo species, it forms an iron 5 oxo species in the heterolytic OO bond cleavage mechanism. Whereas OO bond homolysis leads to an iron 4 oxo species. On the other hand, this superoxide can accept 2 electron and 2 proton, can form iron 2 hydroperoxo species also. And this iron 2 hydroperoxo can undergo OO bond heterolysis to form iron 4 oxo. So this is a mononuclear pathway. On the other hand, in a dinuclear pathway, instead of an electron donor or a cofactor, another iron center can provide the second electron to form a peroxide species. Then it can form a diiron 3 peroxo breached, where the peroxo breached OO bond undergoes homolytic cleavage to form iron 4 oxo species. And particularly for the hydroxylation reaction, cis-dihydroxylation reaction, or epoxidation reactions, these high valent iron oxo oxidants are involved. But as I said, that there are other enzymes where this high valent iron oxo may not be involved. This superoxide, hydroperoxide, or peroxide species may be involved in the reaction pathway depending upon the enzymatic structure, enzymatic function, and definitely for which nature created a particular enzyme. So similar enzymes are called CC bond cleaving dioxygenases. These aromatic or aliphatic CC bond cleaving dioxygenases are involved in the degradation of toxic pollutants. We create a lot of toxic pollutants, but Mother Nature has a machinery to deal with that. And therefore, this could be aromatic CC bond cleavage enzyme or aliphatic CC bond cleaving enzymes. Some aromatic CC bond cleaving dioxygenases include 2-aminophenol dioxygenases, where aminophenol rings get cleaved to form such kind of a product, and then eventually this can undergo a dehydration to form a heterocyclic compound with carboxylate functionality, which is a picolinic acid derivative. Or there is another set of enzymes called gentisate dioxygenase or salicylate dioxygenase or naphtholate dioxygenase. They also cleave the aromatic ring of hydroxy carboxylate substituted aromatic compounds to give ring cleavage products, and also flavonol 2,4-dioxygenases, which are present in many foods, particularly those which we know are antioxidants. And in the process, this particular bond gets cleaved, giving rise to this product, and eventually this can get hydrolyzed and you get polyhydroxylated products which can easily be degraded by soil bacteria, and that would be source for the carbon and energy for those bacteria. One such aliphatic dioxygenase is CC bond cleaving dioxygenase is 2,4-dihydroxyacetophenone dioxygenase. And this is primarily, I'll talk about this enzyme today. This is an aliphatic CC bond cleaving dioxygenase, which cleaves the aliphatic CC bond of the hydroxy ketone in the presence of oxygen as the terminal oxidant, and this carboxylic 4-hydroxybenzoic acid is formed in the reaction, and another product is formic acid. And this particular enzyme is involved in the formation of this carboxylic acid, and this carboxylic acid later utilized for the formation of esters and other things by different plants. And I will show you that why this particular enzyme we picked up, because this is easy to work with, not the enzyme, but with this kind of substrates like alpha hydroxy ketone, because they can coordinate to the metal center through this carbonyl oxygen and the hydroxyl oxygen. And then one can form ternary compound, which are easy to study spectroscopically, easy to crystallize, and definitely easy to perform the reactions and mechanistic studies. Why this particular enzyme we were interested in, and where this enzyme, how this enzyme works. So this enzyme is actually involved in one of the degradation steps of very toxic compounds from plastic, is called bisphenol A. And here you see the bisphenol A. Bisphenol A initially get hydroxylated by soil bacteria to form this hydroxylated product selectively on this methyl group. And once this is done, then it can undergo further reaction, rearrangement reaction to form this product, which effectively form an alpha hydroxy acetophenone, 2,4-prime dihydroxyacetophenone. And this is the end product, which can get cleaved this CC bond by the enzyme dihydroxyacetophenone to form benzoic acid and formic acid. So we were interested in this particular enzyme because we wanted to show the power of synthetic inorganic chemistry. That even without much understanding of enzymatic system, synthetic inorganic chemistry can provide insight about the enzyme's mechanism and structure and reactivity. So when we started this project back in 2011, there was almost no information about this enzyme. Few informations were there. The active site structure of the enzyme was not known, but the sequence homology of this enzyme isolated from a species called allyl species, which indicated that this is a member of cupin superfamily of proteins. What is a cupin superfamily of proteins? Basically, cupin superfamily consists of a 6-stranded beta barrel fold. So it's more like a barrel, and it has 6-stranded structure, and it has normally for the cupin superfamily of enzyme, you have 3 histidines in the active sites. So the structure of the DAD was not known, but another enzyme in this cupin superfamily, which I have shown here, is called gentisec 1,2-dioxygenase, was known. And it was known that that particular enzyme has this kind of a structure where the active site metal ion was coordinated by 3 histidine residues, and the other face of the octahedral were occupied by labile water molecules. So anticipating that since DAD is a member of cupin superfamily of protein, the similar structure is anticipated, and therefore we wanted to test this. And there was almost no information about the mechanism. What was known from a biochemical study of this enzyme, that this is an oxygenase enzyme, where oxygen atom from O2 molecules are incorporated each product or each carboxylate product. So we thought of, okay, let us work on this, since this is a cupin family of protein where you have 3 histidines. Therefore, we thought of making molecules with 3 nitrogen donors ligand, which is a tris-pyrazolyl borate. And these 3 nitrogen donors from 3 pyrazole rings bind to the metal center in one face of the octahedron. Now, if we can utilize this particular labile groups XX of the ternary iron 2 compound with a model substrate alpha hydroxy ketone, still there will be 6 coordination site vacant for the reaction with oxygen. So keeping that thing in mind, we prepared the first compound of this tris-pyrazolyl borate ligand, which we abbreviated as TPBH2, because each phenyl ring, the 3 and 5 position on the pyrazole ring were substituted with phenyl groups. There was a reason. I will discuss that in question answer session. What is the role of those phenyl groups? And we used model substrate instead of 4-hydroxy acid, 2,4-prime dihydroxyacetophenone. We used just 2-hydroxyacetophenone as a model substrate. And also we used another substrate called 2-phenyl 2-hydroxyacetophenone. So instead of hydrogen here, now we have a phenyl group. And we synthesized the iron 2 compounds of the ternary compounds by reacting the ligand with iron salt and the deprotonated form of these hydroxy ketones, and we got the ternary compound TP-phenyl 2 iron 2 HAP hydroxyacetophenone and TP-PH 2 iron 2 BHAP, which is for this compound. As you can see from the optical spectrum, there is a charge transfer transition in the visible region between 450 to about 600 nanometer, and this is typical of a metal to ligand charge transfer transition. So metal to ligand, because iron is in +2 oxidation state, and here you have a ketonic carbonyl which has a pi star, vacant pi star. So where metal to pi star orbital of the keto group charge transfer transition can take place, and which is observed in the optical spectrum in the visible region. And finally, we could crystallize that compound. You see here, this is a 5-coordinate iron 2 compound, and the bond lengths are clearly indicative of a high spin iron 2 compound. This is where this particular hydroxyacetophenone binds in a bidentate fashion as we anticipated. And one might consider, or one might even think, that since it's an alpha hydroxy ketone, it's likely that hydroxy ketone can undergo enolization for extended conjugation, but from the crystal structure, it's very clear that the alpha hydroxy ketone binds in an alpha hydroxy keto mode, not in its enol mode, as is clear from the carbon-oxygen bond distance CO1 as well as this CO2 bond. So this 5-coordinate compound was then utilized to study its reactivity towards oxygen. This compound reacts with oxygen very, very fast, and therefore, but there is an unfortunate part is that because of the presence of so many phenyl groups on the ligand backbone of the compound, this is not soluble in many polar solvents. So we had to initially work in benzene, and then eventually we worked out that some other solvent can be used, which I will cover in my subsequent slides, as you will see. This compound reacts with oxygen at 10 degrees Celsius because at room temperature, the kinetics is very, very fast. So it's not easy to monitor by diode and spectrophotometer. And at 10 degrees, you can see that during the reaction with oxygen, the charge transfer chromophore from the iron 2 to keto charge transfer transition slowly decays, and eventually it gives you a featureless optical spectrum. And you see here that the mass spectrum of this solution shows that the ligand remains intact. Now it's an iron 2 compound. So if we started from iron 2, we end up with iron 2, but the charge transfer chromophore is gone. So what is happening there? So we initially was pretty hopeful that definitely the CC bond of hydroxyacetophenone would cleave as we expected, and that would give you benzoic acid, and that benzoic acid where the benzoate anion can then remain coordinated to the iron center, and we therefore, prior to the reaction, we prepared this iron 2 benzoate compound, and we characterize this compound structurally as well as by different spectroscopic techniques. And here is the NMR spectrum, paramagnetically shifted proton NMR for the typical for the high spin iron 2 compound, as you can see here from -30 ppm to about 60 ppm. This is for the iron 2 hydroxyacetophenone compounds. But at the end of the reaction, this spectrum changes to this spectrum. And this spectrum matches perfectly with an independently synthesized iron 2 benzoate compound. And therefore, we found that after analyzing this NMR spectrum, as well as analyzing the organic products after acidic workup, because if we add acid and if we destroy the compound and then extract benzoic acid to organic solvent, we could found there is no trace of hydroxyacetophenone, but it's purely a benzoic acid. We also tested formic acid by a different technique. So therefore, quantitative transformation of hydroxyacetophenone to benzoic acid and formic acid took place in the reaction. Now, instead of hydroxyacetophenone, when we took this particular compound, which is a benzoane, what we call it, this also reacts with oxygen very fast. Similar optical spectral features, the kinetic mode, a first order decay of the chromophore took place. And after analyzing the product in this case, we found, because it is expected that if this CC bond gets cleaved, you expect 2 equivalent of benzoic acid. And that's precisely, you don't have any other product. You don't get formic acid or anything. So here for benzoic acid is formed in this reaction. But interestingly, the yield is not, or conversion is not quantitative. 85% conversion took place to benzoic acid. Interestingly, we got another product, which is an auto-oxidation product of this compound. We all know that in the presence of metal salt, this alpha hydroxy ketone converts to this benzyl. This is 1,2-diketone product, and we got about 12% conversion for this. And we have run this experiment several times. We found a very similar results in the error range of ±3%. Then we wanted to see how this reaction is taking place, why there is a difference in the reaction. So we wanted to study a leveling experiment. And when this reaction was performed in the presence of O16, hydroxyacetophenone gives you benzoic acid. And this benzoic acid reaction mixture, after treating with diazomethane and extracted the methyl ester, it shows up an ion peak in the GC mass spectrum at 136.12. Whereas when the same reaction was carried out with isotope leveled 18 oxygen isotope leveled O2, and the reaction mixture after treatment with diazomethane, it shows a 136 peak as well for the methyl benzoate. But in addition to that, there is a peak at about 138 here, you can see. So when we analyzed the ratio of normal O16 and O18 product, we found that about 40% O18 is incorporated into benzoate, not 100% as predicted in the original paper in the biochemistry back in 1999. And then we proposed that this is something like this CC bond is getting cleaved, benzoic acid is forming, where partial incorporation of oxygen took place. And similar observation was also seen in case of the benzoane compound. And here also partial incorporation took place, 58% incorporation of O18 took place into benzoate. So 136 peak is there, 138 peak is there, and there is a very small peak at 140. We'll talk about this when I talk about the mechanism for this reaction. Now, when the same reaction was carried out with normal O16, but in the presence of water, where the oxygen atom of the water molecule is 18 isotope level, we do see a little incorporation of oxygen atom from water as clear from this 138 peak. And it's about 10 to 12% incorporation of O18 into benzoate took place. So overall, this reaction, when we reported this back in 2012, and this work was done by Shanthan, who is now assistant professor at IIT Delhi, we first reported the biomimetic functional model of this enzyme. And we predicted that iron must be in iron 2 oxidation state in the enzyme. And the iron must be coordinated by 3 histidines, just like 3 nitrogen donors. So we predicted what would be the structure and what would be the reaction and the mechanism for this reaction. We tried to do an interception of the species, trying to understand the initial mechanistic studies indicated that if we use phenol in the reaction medium, which is a typical superoxide radical scavenger, we do see formation of this coupled product formed via this phenoxyl radical from 2,4-ditertiary butyl phenol. And when 2,4,6-tritertiary butyl phenol is used, this phenoxyl radical is formed. And as you can see, is a typical spectral feature of the phenoxyl radical. And you now have an EPR spectrum typical of the radical species that is observed due to the formation of this phenoxyl radical. And eventually this phenoxyl radical then decays and it forms this quinone product. So the model compound here, in the presence of an external phenol substrate, oxidatively cleave the OH bond homolytically. Now, what happens to the substrate? If I add excess amount of phenol in the reaction, in fact, the coordinated alpha hydroxy ketone does not show CC bond cleavage reaction. That means initially the active oxygen species, which is forming on the iron center, is more prone to do intermolecular hydrogen atom abstraction rather than intramolecular oxidation of the alpha hydroxy ketone substrate that is coordinated to the iron center. So what we proposed in this communication paper in 2012, that an iron 3 superoxo species is initially forming, and then it cleaves this superoxo species cleaves the weak CH bond of phenolic substrates like 2,4-ditertiary butyl phenol or 2,4,6-tritertiary butyl phenol. But in the presence of, in the absence of anything, it reacts with this substrate through a mechanism which I'm coming in a moment. It cleaves the CC bond of that hydroxy ketone. Now this cis hydroxy ketone compound is important that this hydrogen atom of the substrate is essential. That indicates that this superoxide likely interacts with this CH bond, because when a substrate or substrates devoid of alpha hydrogens were used in the reaction, no CC bond cleavage products were observed. And therefore, we proposed that an iron 3 superoxide species initiated the oxidation of CC bond cleavage reaction. So then we came up with this proposal that iron 2 compound with coordinated alpha hydroxy ketone reacts with oxygen to form initially an iron 3 superoxo species. Now we proposed that it reacts with phenol, or in the absence of anything, this can abstract this hydrogen atom. But there is a possibility that this iron 3 superoxo species, which is relatively electron rich, can attack the carbonyl carbon, because there could be a nucleophilic attack at this carbonyl carbon. So that possibility cannot be ruled out. But if we just see that, okay, if this superoxide anion attacks the carbonyl carbon, it is expected that it would form this peroxide species. And this peroxo species then can undergo rearrangement via OO bond heterolysis, and in the reaction, it is expected that aldehyde would form as one of the product. And the iron complex would form an iron 4-oxo benzoate species. And this iron 4-oxo is very, very reactive. This can in fact attack this phenyl group nearby, but in the reaction, we do not see such intra-ligand hydroxylation or the formation of aldehyde. And therefore, we discarded this particular pathway is operational. So then we considered that probably the alpha hydrogen atom, because we have shown the importance of alpha hydrogen atom in the CC bond cleavage reaction, therefore we thought that, all right, this alpha hydrogen atom could be abstracted by this superoxide anion with the formation of this iron 3 hydroperoxo species with a metal bound ketyl radical, which can then transfer an electron to the iron center, can form an iron 2 compound along with the CC cleavage product benzoate. But in this pathway, we have seen that low incorporation of labeled oxygen into carboxylate, 40 to 50% oxygen atom from O2 is incorporated. If this is involved in the reaction pathway, what are the intermediate steps that are involved, which can explain the experimental result of low incorporation of labeled oxygen? To understand that, we subsequently carried out the reaction of mechanistic studies. But in the meantime, after our work was published in 2012, as you can see, the crystal structure of this enzyme was solved. And as you can see in this paper by Cooper's group in 2014 and subsequently in 2015, with a very high resolution crystal structure, they said in their paper that this compound, means our compound, was an analog of the substrate 2-hydroxyacetophenone and the crystal structure, and their structure showed that this model complex has many of the key features that are expected to occur at the active site of the enzymes, such as direct coordination of iron to the 3-pyrazole ring and both oxygen atoms of the alpha-hydroxy ketone group. And intriguingly, this compound was found to react with oxygen, yielding the expected products benzoic acid and formic acid. And Paria et al. Proposed that oxygen binding leads to the formation of an iron 3 superoxo species, which abstracts an hydrogen atom from the alpha carbon of the alpha-hydroxy ketone. And they accepted, in fact, after the biomechanistic studies on the biological system, they accepted that whatever we proposed is correct. But we were not happy. Why? There was low incorporation of oxygen atom from O2. What is the reason? So we wanted to test this further, and therefore we used a different alpha-hydroxy ketone substrates to explore if electronic parameters or electronic effect had any role on the reactivity. So we used different model substrate like this 2-hydroxycyclohexanone or this electron donating methoxy group on the alpha-hydroxy ketone substrate of benzoin or a chloro-substituted substrate. And we prepared their ternary iron 2 compounds. This is the crystal structure of alpha-hydroxy 2-hydroxycyclohexanone coordinated 5-coordinate iron 2 compound. And here again confirmed that this is this binds with the keto group and the hydroxyl oxygen, where the cyclohexyl group here adopts a chair conformation. Then we studied their reactivity, and as you can see here, the reactivity trend. The electron withdrawing group here, the chloro-substituted substrate, coordinated to the iron center, reacts much faster in the order of 10 to the power minus 2 second, 3.8. All are in the same range, these two. This is the benzoin. I have shown that earlier. Electron donating methoxy substituted one reacts much slower, and the cyclohexyl substituted form reacts even slower. The product analysis from different substrates reveal that for this one, since this is a cyclic compound, now you get a dicarboxylate product from hydroxycyclohexanone. But in this case, just as we observed for this particular substrate benzoin, here also we see about 13% of diketone formation, although the major product is CC cleavage product, dicarboxylic acid. In this case, we see that here 18% diketone product is formed, but when we used chloro-substituted substrate, here this particular substrate, no auto-oxidation 1,2 diketone product was observed. Here only the chloro-substituted benzoic acid and benzoic acids were formed in 1:1 ratio. And this tells that definitely this electronic effect at this keto group plays a role. So we wanted to test their reactivity and the mechanistic studies by leveling experiment. And the cyclohexyl substituted substrate here shows that when the reaction carried out with isotope leveled oxygen, we do see peak for the ester of the dicarboxylic acid. Shows ion peak not only at 405, because 405 is this particular ester of phenycyl ester of benzoic or dicarboxylic acid. As the ion peak is seen at 405.16, but when the same reaction was carried out in O18, we do see peak at 405, 407, 409, and even we do see peak at 411. Here in this case, cyclohexane 1,2 dione is formed, which shows an ion peak at 112 as a part of the fragment. And when the same reaction is carried out with isotope leveled water, in addition to 112 peak, 114 peak is also observed. Now we understand the loss of leveled oxygen into the carboxylate product, but how the oxygen atom is incorporated into this diketone product. This was a surprise. And when you have surprise, that means now you have a lot of opportunity to explore that. So we found in this particular case that alpha hydroxy ketone gives you a carboxylic acid and a diketone where one of the ketonic oxygen is partially incorporated when water is added in the reaction medium, as clear from the mixed leveling experiment using 18 oxygen leveled water. Then we went back to the original paper in 1999, where they have said that the reason for this leveling is not clear because they have not done a detailed mechanistic study on the enzyme. And moreover, they have shown that it is, however, possible that there may have been some exchange between H2O18 and the substrate during the course of incubation, but this has not been tested. So we thought that, is it something that loss of oxygen is taking place during the reaction pathway, or oxygen is incorporated during incubation prior to the reaction with oxygen? Because you have a carbonyl group there, which is coordinated to the iron center, a Lewis acidic iron center. It can allow the exchange of the oxygen atom of the keto with water molecule. So therefore, we wanted to see that. But the problem of so far with the ligand that we used, because the ligand was monoanionic. So the final product, where the benzoate coordinated iron 2 compound makes this whole complex neutral, and therefore they do not fly well as a whole molecule in the ESI mass spectrometry. So therefore we wanted to find out that how we can make a complex where from where we get a final product which must be monocationic, so that the cation can fly in the ESI positive ion mode, and we can unambiguously see that whether the leveled incorporation is correctly assigned. And therefore we used this particular ligand, tetradentate ligand, not only to see this thing, but also to check why Mother Nature adapted 3 histidine residues, not 4 nitrogen donating 4 histidine residue or some other coordination motif at the active site. And we again used this particular substrate and this substrate and synthesized the corresponding ternary compound here, 6-methyl-3-TPA iron 2 PHAP and 6-methyl-3-TPA iron 2 HCH positive. This is a monocationic species, and the product from the CC bond cleavage of this one would be an iron 2 coordinated benzoate, which should be a cationic species, so that we can test the level incorporation more clearly, and that would give you more perfect result. And here are the compounds. They react with oxygen, albeit slowly compared to the tridentate ligand complexes. And here you see that this also shows the 6-methyl-3-TPA iron 2 benzoane compound also shows CC cleavage product. But the major product is 1,2-diketone product. It is expected because this is a 6-coordinate compound. There is no band coordination site at the iron center. So the compound must undergo a kind of a solvent exchange, or multiple species might remain in equilibria, so that it creates some room for oxygen to react with the substrate. Similar reactivity is also observed for the hydroxycyclohexane system, although we get about 50% dicarboxylic acid and 37% diketone product. Then we wanted to see whether the compound reacts with oxygen and give you, so this compound is an iron 2 coordinated compound, it's a monocationic species. Before it was exposed to oxygen, under inert atmosphere, when mass spectrum was collected, this shows an ion peak at 599. And under inert atmosphere, H2O18 was added, it shows ion peak at 599 and 601. That means that even before the reaction with oxygen, some amount of water is being exchanged with this ketonic carbonyl group. And here, this compound, when react with oxygen, this shows an ion peak at 105, the fragment. And in the presence of H2O18, in addition to 105 peak, we do see a peak at 107, and about 15% incorporation of O18 into diketone took place. So now if we go to that, what happens with O16? With O16, this shows an ion peak at 509, with perfectly isotope distribution pattern for this ligand, iron 2, and benzoate compound in the monocationic form. Same reaction when carried out in the presence of isotope level oxygen, purely 99.9% isotope level oxygen. Here you see that 509 peak is there along with 511 peak, which is a major peak, indicates that 70% incorporation of O18 into benzoate took place. So that means now I have 30% loss at this point. But what is the origin of this loss of 30%? That can be explained from H2O18 experiment. And here we see now 20% incorporation of oxygen into benzoate. Still, I have a mismatch of 10%. Now, if I go back, where this particular species is coming from. So it's about 15% incorporation means this mismatch can now be explained that this is happening during the reaction with H2O18 prior to, or sorry, it's actually taking place in the reaction with oxygen, in the reaction with water, sorry, prior to exposure to oxygen. So this somehow explains the problem that why this loss was. So therefore we came up with the reaction pathway that this alpha hydroxy ketone substrate in the presence of water, because you have always some amount of water in the solvent, because in all these tetradentate ligand system we used acetonitrile. So acetonitrile contains trace amount of water, and this trace amount of water first exchange with this alpha hydroxy ketone before it reacts with oxygen. This compound reacts with oxygen to give you a carboxylate product with partially incorporation of labeled oxygen, but no incorporation of labeled oxygen into the diketone product. But if you use H2O18, it can exchange with this one. So now we already have a partially labeled oxygen in the molecule itself, because they can be in equilibrium. And then when this reacts with oxygen, this gives you benzoic acid with labeled oxygen and a diketone product with some amount of labeled oxygen incorporated into it. And that clearly tells us how this reaction is taking place, and that can also explain why in enzymatic system initially they thought of it's a purely a pathway which is independent of any water exchange, but subsequently they also saw this water exchange, but no explanation was there. And here we now have the explanation and today got from the model compound. So here, this superoxo species, now we propose that it abstracts the hydrogen atom and it forms this iron 3 hydroperoxo species. And then eventually this goes to CC bond cleavage product, which I'm coming in a moment with details mechanism of the CC cleavage. But when this one, the electron is transferred to the iron center, this can form an iron 2 hydroperoxo species, because iron 3 center gets 1 electron reduced. Now this will form an iron 2 hydroperoxo species and form 1,2 diketone product. So this is a pathway which is operational by transfer of electron to the metal center. Whereas CC bond cleavage takes place with the involvement of this iron 2 hydroperoxide by interacting with this ketyl compound. Now the question is this at all formed in the reaction? Because we have seen that in case of tetradentate ligand system, this diketone is the major product in some cases. And if it is so, this would be the metal coordinated iron oxygen oxidant. Can this OO bond undergo cleavage, or can this participate in the substrate oxidation? Because if I have a good amount of this hydroperoxo species or its OO bond cleavage product, iron 4 oxo or whatever it is, then that must oxidize some substrate, and therefore externally, if we add substrate, the substrate must get oxidized without compromising the CC bond cleavage pathway. So we wanted to see if that can happen, and whether spin state also matter, because all the compounds I talked about, they were high spin iron 2 compound. In case of tridentate ligand, CC cleavage was the major pathway. In case of tetradentate ligand, it was more or less 2 electron oxidation of hydroxy ketone that was major pathway. But here, if we now use a ligand, which is a very strong field ligand with 3 nitrogens and 1 carbon ion, and this
now we are working on a manuscript now on this, and this particular ligand, Partho Haldar, who is in the audience, who has started working on this ligand system, where he has shown that this can stabilize iron in the low spin state. And Sridhar recently isolated this alpha hydroxy ketone compound and allowed to react with oxygen at room temperature in acetonitrile, and found here, in this case, 100% 1,2-diketone product takes place without any cleavage product. So that means that I can have either this species or its OO cleavage form iron 4-oxo. And indeed, we could see recently the formation of such iron 4-oxo species, which has been characterized by different spectroscopic tools, including Mossbar spectroscopy, but I will not get into that, where it is saying that spin state is equally important. A low spin compound will not show CC bond cleavage product. Just we have seen in an enzymatic system, the iron center is in the high spin state. So the ligand denticity, to summarize, we have seen that a tridentate ligand cleaves the CC bond of hydroxyketone quantitatively when I used alpha hydroxyketone hydroxyacetophenone. In case of benzoin, it gives you a little bit of 1,2-diketone product. In case of tridentate ligand, you see the diketone product formation pathway is actually a minor pathway. Whereas for tetradentate ligand system, diketone formation pathway, at least for this one, is the major pathway, whereas CC bond cleavage pathway is the minor pathway. So therefore, complexes of the tetradentate ligand, they afford higher amount of diketone product. And in the case of tridentate ligand, it is the CC bond cleavage products which is primarily playing a major role. So overall, the mechanistic proposal that Rubina came up with as a part of her PhD work. Rubina now is teaching in Krishnanagar College. I don't know whether she is in the audience. And this particular compound, as so far, we have shown that this is involved a common intermediate which can diverge into two different pathways, either in 1,2-diketone formation pathway with the formation of iron oxygen oxidant that can oxidize substrate and can decay. In the CC bond cleavage pathway, this iron 2 peroxo species can form, and this peroxo species then can undergo a Kriege type rearrangement with the formation of this lactone with metal coordinated hydroxide. This metal coordinated hydroxide can exchange with water. So not only that water can exchange with the initial iron 2 compound, but also during the reaction pathway, water can exchange its oxygen with the oxygen atom of this species, forming an iron 2 hydroxide compound along with this ester. Now a metal coordinated hydroxide is a very reactive. It can attack this particular group, forming the carboxylate product, or it can also attack this with equal probability. So when hydrolyzed, it can form an iron 2 coordinated carboxylate compound with partially leveled oxygen, which can now be explained by this mechanism. As I have shown you in one of the compound, where not only that we got one oxygen atom incorporation, but also two oxygen atom incorporation with 36 peak. We also had a little bit of 140 peak for the methyl ester of benzoic acid, can be explained from here. So see, if we consider that this species now undergoing hydrolysis, this can attack this carbon and this can attack this carbon. So when it forms a benzoic acid by attack of this hydroxide on this one, now you see both the level oxygen products would be there. That means two oxygen atoms will be leveled oxygen, and as a result, two leveled oxygen will be incorporated, and that is reflected into the benzoic product, what we have seen, the 140 peak. Interestingly, after this mechanism that we said that water is a very, very important in this reaction pathway, this mechanism we proposed in 2015. Then a group came up with a mechanism through quantum mechanics and molecular mechanics studies. They found that they said that there could be a different pathway, although they found that the mechanism, what we proposed is one of the pathway, although they came up with a completely a different pathway. But what we felt that this is less likely because our experimental result, particularly the leveling experiment data, could not be explained by could not explain this mechanism. Then recently, my colleague Professor Ankon Pal and Professor P. Manjana, they came up with an interesting quantum mechanics and molecular mechanics and investigation, and they found that one of the water molecules in the active site plays pivotal role in directing the CC bond cleavage pathway. And as a result, they found that the mechanism, what we proposed in our 2015 paper, turns out to be correct. And they found that is the main interesting point. And they also have found that the transition state, the peroxide species, is the active species, which eventually goes to the CC bond cleavage pathway. So with this, I think my time is 2 minutes left. So I would summarize by saying that we have developed functional models of 2-hydroxyacetophenone dioxygenase, which cleave this iron complex, just like enzymatic system, cleaves the CC bond of metal coordinated alpha hydroxy ketone, and the mechanistic studies, reactivity studies provided useful insight into the enzymatic reactions even before the enzymes mechanism was known. So that is something is a pleasure, and here we stand now that a simple synthetic inorganic chemistry can in fact provide a lot of useful information about enzymatic reactions, and that is precisely the satisfaction as a researcher. So with this, I will end up, and now I will go to the last slide, and the most important slide, I would say, particularly because it was very easy for me to present this work, but this is a result of very hard work by a beautiful, group of beautiful students from in my group, very intelligent, hard-working people who always work with in my lab, and their effort eventually brought us here. So this work was initially started by Shanton back in 2011. And then this project was picked up by Rubina, who finished PhD a couple of years ago. And then Sridhar partly is involved in this project as the last work that I talked about. And the other members also in the group contributed some way or others, or at least, keeping a beautiful environment in the laboratory. Finally, I'd like to thank the funding agencies. As you understand, this work involves a lot of money. And money came from the Department of Science and Technology through Science and Engineering Research Board, Indian National Science Academy, Council of Scientific and Industrial Research, and Swedish Research Council. So with this, I will end up, and I thank you all for your attention, and I'll be very happy to take up questions and comments on this. I think I am on time.
Yeah, thank you, Professor Pine. So now is the time for questions from the audience.
Thank you, Subhasis.
If anybody has any specific question, please put on.
Yes, please put on in the chat box.
Yeah. It's difficult also, takes time to type, no?
Yeah. Afterwards, you can say all this. Yeah, this is one such.
Sir, one question appears.
There is one question that I can see.
Yes.
Let me see the chat box full. Yeah. Why is this reaction so fast at room temperature? Okay. So this question came from Kriti Alam. It's an interesting question. As I have shown you that, first of all, these iron ternary compounds are not only coordinated by an anionic ligand, because 3-spirazole borate is an anion. And moreover, the alpha hydroxy ketone is in monoanionic form. So both two anionic ligands are coordinated to the iron center. Obviously, your iron 2 reduction potential will drop, and absolutely they are very easy to react with oxygen. But again, there is another one, as you have compared, and I already told that compared to the 6 coordinate compounds of the tetradentate ligands, this tridentate compound reacts faster. The reason is that now in case of a tridentate ligand, you have a 5 coordinate compound, unlike the 6 coordinate compound that we get using tetradentate ligand. And that is the reason why they are faster in terms of their reaction towards oxygen. This is clear to you?
Yes. Yes.
Yes.
If is there any question, please write in the chat box. Second question appear. Hello, sir. Hello. Hello.
Yes. What is that question? I cannot.
Yes, sir. Second question
Yes,
appear
yes.
in the chat box.
Hello. Yeah, I got it. I
Yes,
got
sir.
it. What is about the partially selected atom within? What is about the partially selected atom within a molecule? I did not get the question.
Right.
What is about the partially selected atom within a molecule? Shreyoshi Jana, could you please
Complete the question, sir.
and tell what exactly you wanted? Does it mention the atoms to be radioactive or not? No, they are not radioactive. You see, 18 isotope level, they are lighter elements. They are not heavy elements, and therefore they are not radioactive. The isotope level oxygen is it's safe, easy to work with, and it's not that expensive, although it is quite expensive. I will not say it's cheap, but it's not very expensive also. And normally our customs people do not create much problems. So therefore I think it's easy.
Hello. If
Yes.
hello, Devasri Rai sir.
Yes, okay. So
That if there is no question,
concluding here. Okay.
yes, sir.
Professor Pine, I have a very small query for you.
Yeah, I'm waiting. I'm waiting for your question.
Okay, so that trispirazolyl borate,
Yeah.
you have the phenyl substitutions
Yes.
on the pyrazole
Yes.
backbone on the
Yeah.
both sides. If you go for dimethyl substitutions, the dimethyl
Yeah.
pyrazole borate, what is your expectation?
Okay, that's it. Yeah.
What is your expectation?
Okay, so the problem is that why, as I said at the beginning, that I would tell you about this, but somehow during the mechanistic studies I did not talk about it. That if you take the methyl substituted one, the problem is that the methyl groups on the pyrazole rings, that they do not engender sufficient steric bulk, and as a result it forms a homoleptic peace complex, and therefore it is a fatal flaw. You do not get ternary compound. So we can get it using isopropyl substituted one. You can synthesize that tertiary butyl substituted one, but since the tertiary butyl substituted one is sterically too demanding, oxygen cannot come close to the iron center, and therefore they remain very, very unreactive.
I get it now. So, Professor Pine, the thing is that if you go for this methyl substitution or no substitution, at one point of time, can you expect that the iron center can be octahedral also? That means it can
because the iron center will then come.
That you need the distortion. The huge distortion.
Right, absolutely. So the problem with the methyl group, I said, is that it forms bis complex as you always isolate, but that does not necessarily rule out the formation of a ternary compound with metal coordinated alpha hydroxy ketone in solution. So there could be a dynamic equilibrium. There could be dynamic equilibria in solution where you have a metal coordinated hydroxy ketone as well as the bis complex can form. But the problem is that if thermodynamics, thermodynamics always lead the whole reaction towards the homoleptic complex, and as a result, CC bond cleavage activity is not observed in the reaction. So it's a thermodynamic stability of the homoleptic complex that drives the reaction towards that, and as a result, a very little amount of hydroxyketone remains coordinated at a given point of time for reaction with oxygen.
Okay. Thank you very much. Okay, so let us conclude here for a very good presentation from Professor Pine. So thank you.
Okay. So I have a question. I actually got a question. If it is not too late, I must answer this question. Why is the oxygen atom partially colored? Just to indicate that it is a leveled oxygen from 18. That is the reason. Okay, so that people can understand that this originates from the leveled oxygen.
Yes.
Okay, now Tithi.
Can ultra-fast dynamics be utilized for studying the reaction at room temperature? We have not done anything, and these are paramagnetic systems, so I do not know whether that can be done. I don't have any answer to this.
Okay then.
Okay.
Let us thank Professor Pine for a beautiful talk.
Yes, sir.
Thank you very much.
Thank you, sir. Thank you both of you, sir, to session chair Professor Devashish Roy and Professor Pine for joining this webinar. I am very grateful to both of you, sir. Thank you, sir.
Thank you, Tithi.
Thank you, sir. Thank you, sir. Professor Pine, sir. Hello.
Thank you very much. Thank you, Tithi.
Thank you, sir.
Yeah, thank you. Thank you very much. Yeah, thank you very much.
Okay, sir. Okay. Now, we are going to the paper presenting section. And in this session, I request Professor Amiyo Kumar Panda, Professor, Department of Chemistry and Chemical Technology, Vidyasagar University, to chair the session, sorry. Now, hello, Professor Panda.
Yeah, I am here.
Okay, sir. Sir, actually, at first I call all the name and all the name, and those who are present here, please unmute your phone and give the answer yes. Gyanendranath Maiti. Gajendranath Maiti. Dr. Maiti here. Please unmute your phone and response yes. Dr. Kumar.
Yes, I am present here.
Okay. Dr. Kumar Roy.
Yes, I am present.
Okay. Shanta Kumar Manna. Dr. Sushanto Manna. Dr. Manna, are you present here? Soumya Sundar Mati. Dr. Soumya Sundar Mati.
Yes, yes.
Okay. Dr. Indranil Chakraborty. Dr. Indranil Chakraborty. Dr. Lakshmikanta Das.
Yes, I am present. Yes, I am present.
Okay. Dr. Sip Sundar Das. Shankar Das. Dr. Sip Shankar Das. Shankar Roy.
Present.
Present. Okay. Deepak Kumar Saha.
Yes, present.
Okay. Our hand.
Yes, I am present.
Deblina Sarkar. Dr. Deblina Sarkar.
Yes, ma'am, I am
Okay.
present.
Dr. Aniket Chaudhary. Dr. Aniket Chaudhary. Dr. Bubun Banerjee. Sir. Hello, Professor Panda. Sir, majority are present. I hand over the microphone to you, sir, to continue the session.
Could you please tell how much time is allotted for this?
Sir, 5 to 6 minutes. 5 to 6 minutes.
So, thank you, Dr. Mikey, for giving me the opportunity sharing the session without any introduction. Because we are ending since the morning. So, let me first invite Dr. Bajendra Mikey. Bajendra, are you there? Okay, if not, then may I invite Dr. Pompa Guo and rest of the speakers, please mute your phone. And Dr. Pompa Guo, could you please present your work and your time is 5 to 6 minutes.
Okay, sir. Sure.
I can see Rajendra over there, but I don't see why he is not responding. He was there. Yeah, exactly. Okay, then Rajendra, please, can you present your work? Just go on present.
Sir, I think we should mute our microphone.
Yes.
Yeah, everyone should
Except Pompa Guo and Bajendra Mikey.
Can you present your work? Others, please mute your phone.
Somo Sinaroy, please mute your microphone. Somo Sinaroy, please mute your microphone. Then now Pompa Guho or Gajendra Maiti, anyone can present.
Sir, should I present now?
Yeah, I think Pompa, you present your work before Gajendra starts because Gajendra needs some time. So
Okay.
Dr. Guha, please present your work.
Okay,
So
sir.
time is 5 minutes.
Okay. Can you see my slides? Hello, can you see my slides?
Yes, ma'am.
Hello, can you see my slides?
Visible. It's visible.
Yes,
it is visible. You can
Okay, I'm starting now. Okay. Good afternoon, everyone. Welcome to my presentation. Today, the title of my presentation is Click Chemistry and its application in metal coordination. So first, we need to know what is click chemistry. The word click refers to such an instant reaction as if you just click the shutter of a camera and it's done. So the reactions are really fast. In 2001, Professor Sharpless described some reactions which are of high yield, wide in scope, create easily removable byproducts, stereo-specific and easy to perform. So all those reactions Professor Sharpless told, okay, they can be called as click reactions. So there are major classes of click reactions, but among all the reactions, copper-1 catalyzed 1,3-dipolar cycloaddition of alkyne and azide is an ideal click reaction. Those copper-1 catalyzed reactions are highly regioselective. They only give 1,4 substituted isomer, that is 1,4 substituted 1,2,3-triazole ring. Similarly, the ruthenium catalyzed azide alkyne cycloaddition is also regioselective. It gives the 1,5 isomer. Hence, the Professor Sharpless and his group, they reported the method of the reaction. They used copper cupric sulfate and ascorbic acid as the reducing agent. During the reaction, ascorbic acid reduces copper-2 to copper-1. But in our group, during my postdoctoral stay at Florida State University, we did the same reaction but in a different reaction condition. That is, we used the alkyne and azide and copper-2 acetate as the active catalyst in methanol or tertiary butanol as a solvent. The reactions are even faster and the even more cleaner. I mean to say, it's more easy to purify the product and the yield is really high. You can just see in the vial, the greenish blue color solution indicates copper-2 acetate solution, Once you just add alkyne and azide into it, within just 1 or 2 minutes, the copper 2 becomes copper 1 and the color changes from blue to yellow. So it indicates that the reaction is finished and the yield is like more than 90%. So this reaction is very, very good. We already established the mechanism and the reaction cycle, but here I am not explaining all those things due to the short time. If you are interested, you can just go through this reaction. Using those procedure, I prepare different type of triazole-based ligands, which are very good ligands to coordinate the transition metal centers. So initially we just used these monoazide compounds, and from here we just get this monotriazole product. These ligands are very, very good, and here is the R means the different functional group. We can change this R group just to introduce the different metal centers for the complex. Similarly, we used diazide compounds and just prepared some bis triazole rings. Yes, you can see we just prepare different type of triazole products using the different functional groups. So our aim was to prepare different type of pockets which will be able to bind the transition metals. That means transition metals can sit inside those pockets. And based on those functional groups, we got different types of interesting structures. So here I can show you the different binding pockets mentioned like 1, 2 like that. So here are some beautiful structures. When you used this ligand, and this is actually very poorly coordinating, so it cannot destroy the core of the copper acetate. When we used copper 2 acetate as the metal salt, so it only replaces the actual water molecules and just coordinate the metal center from the pyridine nitrogen atom. But if you use copper perchlorate, where perchlorate is the comparatively weakly coordinating ligand, you can get this type of mononuclear complexes. Similarly, when we just replaces this benzene ring with the pyridine one, you get a different type of dinuclear compounds.
Dr. Guho, please conclude.
Okay, sure, sir. Similarly, we got some Cuban compounds, which are also very interesting. We got some good publication. We did some ITC treatment to do the thermodynamics of the complexes, and we just studied the magnetic properties which show interferometric behavior. So in conclusion, we can say that this triazole moiety can act as a potential versatile ligand to generate transition metal complexes with interesting properties. I must acknowledge my college and all my PhD supervisor and the postdoctoral guide, and thank you all.
So thank you very much, Dr. Guho, for your nice presentation. If there are any questions, may I request also the participants, when the session is going on, you may post your questions, if any. So I hope Dr. Guho will be happy to answer the questions, if any.
Yes, sure.
Any questions from audience? Please unmute your phone and ask the question. If not, let us thank Dr. Guho for your nice and timely presentation. So, oh no, there is a question. There is a question. Dr. Yes. Asked. Can you see the question?
Yes, sir. I can just let me check it.
Yeah, it's, have you tested the catalytic efficiency?
No, during my I was only interested about the magnetic interactions, actually, how this ligand bridge between the transition metals and how the magnetic property changes. So that was my main target. So I did all the magnetic studies.
Okay. Thank you.
Thank you, sir.
Dr. Prashanta Patro, please mute your microphone.
I have one question. I'm Dr. Shunata Haldar. Would I ask the question?
Yes, ma'am.
Actually, this is a very fast reaction, as you told.
Yes, very fast.
Can you measure the kinetics? Can you follow the kinetics of such reaction?
Yes, ma'am. Yes, ma'am. We have established the mechanism. We did lots of NMR titrations, fluorescence titrations with proper functionalities, and we established the whole mechanism. And it is already published in JSCS. I have just shown the reference. I can give you that one also if you are interested.
Okay, thank you.
Okay, thank you, Dr. Guho.
Thank you, sir.
Gajendra Nath Maiti. Gajendra, are you there to ready your? Are you ready? Okay. Yeah, Gajendra Maiti. Okay, fine. He is from Department of Biology, Ashutosh. Gajendra, please make your presentation.
Yes, sir.
Present now. You just click present now. And
আসছে না স্যার এতে আসছে
yeah, now it's coming. Yeah, it's
Yes.
coming. Don't minimize, and other speakers also, you make your presentation, you keep your presentation open and just go to the present. Okay, Rajendra, please present.
Yes, sir. Stop present.
Rajendra, are you there?
Yes, sir. I cannot.
Sir, you have to open the slide window, add a window, not minimize it.
Yeah, that's what I told you. First open the program.
Gajendra Babu, please close
Yes,
all
sir. Man, press.
actually you close the program totally, then freshly you open all that thing.
Open your presentation first, don't minimize it, and then
Present now.
yeah. Maybe we can go for the next speaker. If meanwhile, let Gajendra. Okay, Dr. Sutapara is there.
Yes, sir.
Okay, can you make your presentation now?
Okay, sir.
Dr. Sutapara, she is from Government Degree College, KCL. Dr. Rai, please.
Sir, can you see my presentation?
Yeah, just it's now coming. And we can say it. Please start. Just go to the slideshow and make your presentation. Sutapa, are you there?
Yes, sir. But I can't.
Sutapa,
Yes.
going to slide show. Go to slide show. In the upper corner.
Slideshow.
Go to slide show.
ওই যে ডান দিকে নিচে একটা স্ক্রিনে ছবি আছে
Yes, complete. Yes.
Okay.
Okay, I'm starting.
Okay.
Good afternoon, everybody. Now I am going to deliver on the topic importance for the determination of organic and inorganic contaminants of effluent water. Water is important for sustaining life on Earth. When we drink water, we remain very much cautious, but during dumping of effluent water, we do not show much sincerity. Now, I shall discuss why should we concern before dumping any effluents. Now, let us understand what is effluent water. Effluent water is nothing but the wastewater which is being discharged. This may be a discharged water from a household, from the industries, from the municipalities, or from the public utilities. The next question is where this discharged water, which is technically termed as effluent, is being dumped on. If you look around yourself, you will find your household effluent is discharged into drains if it is in urban area or to a pond or irrigation field or canals if it is in rural area. Similarly, the effluents are being discharged to a suitable water bodies by all concerned, whether it is commercial or non-commercial. In general, an effluent may be tagged by the common people as bad effluent or good effluent by their biophysical shapes, as you can see in the photos of my slide taken from the different web sources. But this biophysical sensing and accepting the effluent as good or bad is not technically acceptable. So it is desired to test the sample of the effluents. India has classified the water bodies on which an effluent can be discharged into 4 groups, namely irrigation fields, marine coast, inland surface water, and public sewer. The acceptable quality of the effluents for discharging into these water bodies are different and stipulated by the Ministry of Environment and Forest. India needs 700 billion meter cube of water per year as per the estimation made in 2014. Amongst these, 690 billion meter cube are available to utilize from the surface water and rest is from other sources. Domestic consumption requirement is 42 billion meter cube, whereas industrial requirement is 37 billion meter cube. Probably you may wonder that why this 700 billion meter cube is required. The major component is the irrigation requirement, which is above 500 billion meter cube. Now, if you look into the effluent side, the domestic sewages generated is 22,900 million liter per day, whereas industrial effluent is 30,500 million liter per day. If you see the installed treatment capacity, it is only 5,900 million liter per day for domestic sewage and 8,000 million liter per day for industrial effluent. That means 75% of domestic sewage and 40% of industrial effluent are dumped to the natural water bodies without any treatment. Now the question is, why this gap and how to mitigate this? Now, if you look into the current status of the commercial water use, most of the industries and urban municipalities are prohibited from using groundwater and advised to go for reduce, reuse and recycle. The effluent water treatment capacity source are mainly active treatment, which is costly but fast. Passive treatment is cheaper but needs years to treat the water. Indian industries are also lacking a treatment plant designing as per their own requirement. These are the parameters to be controlled prior to discharge the effluent to the water bodies. These parameters are classified in 3 groups: organoleptic or physical parameters, undesirable parameters, and toxic parameters. Undesirable and toxic parameters are mainly the organic and inorganic contaminants. These are the details of the standards. Here are the testing of wastewater. These are the testing procedures for determining the concentration of the 34 pollutants. As you can observe, the cost of testing for physical parameters are much more cheaper than the testing of organic, inorganic pollutants of undesirable and toxic parameters. Thus, industries as well as the municipal corporations are reluctant to go for testing those parameters and only treat the effluent. These are the pictographic presentation, and here are the 2 case studies from 2 different industries of same nature exhibiting different characteristics. There are effluent 1 and effluent 2. Effluent 1 contains effluent 1 is very much poor quality and not suitable for discharging. It contains high TSS, that is the total suspended solid. But effluent 2 is good quality but contains the total suspended solid high. But effluent 1 significantly presence of undesirable and toxic elements demands urgent treatment. But effluent 2, this does not significant treatment is required. So mandatory.
Kindly conclude.
Okay, sir. So this is in conclusion. So these measurements are needed to only then we can wish to have a better water environment for our future generation. Thank all of you for your kind patience, and also I thank organizers for giving me such platform. Thank you.
Thank you, Dr. Raman. So one quick question. One quick question from the participants. Any questions? If not, then let us thank Dr. Roy. And may I now once again invite Gajendra Nath Maiti. Gajendra, are you there?
Sir, hello.
Yes.
Sir, actually, one or two participants trying. Then Gajendra, who was something problem in the okay.
So the next is Sushant Kumar Manna from Bidhannagar College. Sushant Manna, are you there?
Most probably no, sir.
And then Soumya Sundar Maiti. Dr. Deepak Shah, could you please mute your phone? Dr. Deepak Shah, could you please mute? Yeah. Thank you.
Thank you.
Soumya Maiti. No?
Yes, sir.
Dr. Indra.
Yes, sir, I am present.
Yes.
Okay, please present your work.
Okay.
Soumya Sundar Maiti. Soumya, are you there?
Yes, sir.
Okay.
Present now.
Yes. Soumya, at first you.
Yes. Titi, please instruct. Yeah. Please.
A window. Yes.
A window থেকে share.
Yes.
Do you see your? Yes.
Okay.
Yes.
Dr. Maiti, please.
Yes, sir.
Good afternoon, everybody. First of all, I want to thank organizing committee and especially Dr. Titi Maiti, head Department Chemistry, PK College. My presentation entitled "Small Molecule in Double-Stranded Nucleic Acid." So, first of all, aim of my work. The aim of my work is to point small molecule binding mode with nucleic acid. Here, the inspected nucleic acid are DNA and double-stranded RNA, and the inspected small molecule are methylene group MB and naphthalimide derivative NP. Now, first of all, we have to know how a small molecule interact with DNA. First of all, intercalation mode of interaction, where molecule inserted between the base pair of DNA and lengthening the chain. Second one is electrostatic interaction, where the small molecule electrostatically attached with the phosphate backbone of the double-stranded DNA. It is less significant. Number 3, group binding. Here, the small molecule is attached in the vicinity of the double-stranded DNA around the major or minor group. Here, our main interest to differentiate this intercalation and group binding mode using two different drugs. First of all is methylene group MB, which is an intercalative drug, and second one is naphthalimide derivative, which is a group binding drug. This is the absorption study. First of all, we have to discuss this, in which the absorption of NP, that is the naphthalimide derivative group binding drug, and MB, the intercalative binding drug, when the ctDNA is added simultaneously in these two type of drugs, the absorbance in both the cases are decreases. But the mainly change of that is that there is the we have to notice that there is a clear isosbestic point in intercalative binding drug at 605 nanometer. So we can differentiate this with the help of this isosbestic point, and there is another point is binding strength or binding constant value. From binding constant value, we can say that the intercalative binding drug has the binding constant in the range of 10 to the power 5 order, whereas the group binder drug has its binding constant in 10 to the power 3 order. Next, fluorescence spectra. From fluorescence spectra, we can see that upon consecutive addition of the DNA in both naphthalimide and MB, that is the intercalative binding drug, there is a subsequent decreases in this fluorescence spectra, and we can see that there is no differences between these intercalative binding drug and group binding drug. So from fluorescence, we cannot differentiate these two type of binding. Number 3, the circular dichromosome spectra. What is circular dichromosome? Circular dichromosome is that when an optically active molecule is exposed in a circularly polarized light, then in presence of circularly polarized light, optically active compound absorb either right or left rotating polarized light, and as a result of it, we observe the circular dichromosome spectra. In DNA, in case of DNA, we observe the two binding two peak. One is around 275 due to base stacking, and another is 245, which is right-handed helicity of the DNA. And we can see that when we added the group binding drug NP, there is no change in the circular dichromosome spectra, whereas it is the insignificant change. But when we added methylene blue, which is an intercalative binding drug, there is a regular decrease in the circular dichromosome spectra. This is due to the intercalative mode of binding, the MB molecule inserted in the base stacking of the DNA, and as a result of it is the 275 nanometer, which arises due to the base stacking, is distorted, and the helicity also changes, and as a result of it 245 nanometer peak also change. The next study is helix melting study. What is helix melting? Helix melting is that at the temperature where double stranded DNA separated into its single stranded. And from this helix melting study, we can see that the naphthalimide, the group binder drug added, when the group binder drug added, there is no change in the helix melting temperature. That is, the helix melting temperature in ctDNA and group binding drug attached DNA, there is no change. This similar type of helix melting temperature, but in case of the intercalative binding drug, the helix melting temperature increases about 10 degrees centigrade. So from helix melting study, we can also differentiate the intercalative binding mode and group binding mode between these two drugs. The next study is the molecular modeling and docking study, from which we can clearly see that the naphthalimide, the group binder drug, is attached in the minor groove of the ctDNA, and the intercalation drug MB, methylene groove, is inserted between the base pair of the DNA. And
Dr. Martin, please conclude.
And from my conclusion, I can say that there may be some variation in experimental result in case of different probe and different DNA, and this successful comparison between group binder and intercalating molecule. Thank you.
Thank you, Dr. Marty, for your nice presentation. So, questions, if any? Dr. Marty, where did you carry out these experiments?
Sir?
Where did you carry out these experiments?
Actually, I carried out first of all my PhD work, and I have now a project with the collaboration in IIT Kharagpur, and I also carried some experiment in my college laboratory also.
Yeah. Since that was missing, that have been missing in your acknowledgement section. This is my humble suggestion.
Yes, sir. Thank you.
Any other questions?
How do you determine organic matter in water? This question for mine.
No, probably not for you.
Yes.
Not for others. The question is the helix melting temperature procedure general for all the systems? This is for your question. This is for you. Is the helix melting temperature procedure general for all systems?
No, not general for all systems. Mainly it is depend on the different factor. One of them is the salt concentration or buffer concentration. If we vary the buffer concentration, then helix melting temperature on the same DNA differ. And also it differ from DNA to DNA and the percentage of GC and AT content. Here I show here the one is ctDNA, another is human testosterone DNA, and their salt temperature and salt concentration is different. Both the cases. So the actual helix melting temperature is different, but the change in group binding and intercalating binding, which I mentioned that for intercalating binding, the change is about 10 degrees centigrade, but in group binding, it is more or less helix melting temperature of DNA, and the group binder drug with DNA is similar.
Okay, thank you, Dr. Mathi, for your nice presentation. Now, may I invite who next? Dr. Indranil Chakraborty?
Due to some unavoidable circumstances, Indranil Chakraborty informed me he is unable to present. Sir, next, please.
The next is Dr. Lokhikanto Das.
Yes, I am present.
Okay, please present your work. Dr. Lokhikanto Das is from Government Degree College, Kharagpur. Dr. Das, please.
Lokhikanto, first open your PowerPoint on the window. Can you hear Lokhikanto? Hello?
Yes.
You first open your PowerPoint on the window, not minimize it.
Okay.
Yes, thank you.
Can I start?
Please. Dr. Das, please make your presentation.
Sure.
Yes. Good afternoon to everybody. Now I am going to present my paper entitled Catechol-like activity of two oxime-based tetranuclear nickel-2 complexes. First I discuss the catechol-like. What is catechol-like activity? Catechol-like activity is the process of oxidation of a wide range of orthodiphenyls, which are commonly known as catechols, to corresponding orthoquinones, and the process is catalyzed by the natural enzyme catechol oxidase. In this scheme, I have shown the oxidation process. Now, catechol oxidase is a copper-containing type 3 active site enzyme, which are usually encountered in plant tissues and in some insects. This is the crystal structure of catechol oxidase, which was first reported by Krebs and his co-workers. The active site of the catechol oxidase contains a dicopper core in which both copper ions are surrounded by 3 nitrogen atoms from histidine residues. Now I discuss the synthetic models of catechol oxidase. Actually, the catechol-like activity of some model coordination compounds have been a topic of recent interest for the development of new bioinspire catalysts. The catalytic action of many dinuclear copper-2 complexes have been developed over the past few decades, along with a number of manganese 2,3 and cobalt 3 complexes. Recent investigations have shown that some dinuclear nickel-2 species can also mediate such catechol oxidation. Thus, the aim of the present work is to synthesize several nickel-2 complexes in order to investigate the catechol-like activity. Here I have shown the synthesis of two tetranuclear nickel-2 complexes from similar oxime-based C-base ligand in an alkaline medium. These two are the crystal structure of two synthesized tetranuclear complexes. Both the complex contain 4 distorted octahedral nickel-2 ions which are held together in a rhombic chair arrangement by the 2 central hydroxido and 4 peripheral oxido bridges. In this slide, I have discussed the catechol-like activity of the synthesized complexes. In this regard, I have chosen 3,5-ditertiary butyl catechol as a substrate, and the 2 synthesized complexes have been allowed to react as a catalyst in this process. In this reaction scheme, I have shown the catalytic behavior of the tetranuclear complexes towards the oxidation reaction of 3,5-ditertiary butyl catechol to corresponding orthoquinone through the formation of substrate catalyst intermediate. Actually, the reaction were determined spectrophotometrically by measuring the absorbance of quinone band at around 400 nanometer as a function of time. The oxidation reaction follow first order kinetics at lower concentration but follow saturation kinetics at higher concentration. The observed rate versus substrate concentration data were then analyzed on the basis of Michaelis-Menton's enzymatics approach in order to obtain the double reciprocal Lineweaver bar plot.
Dr. Das, can you conclude this?
Yes. From this plot, one can easily calculate the turnover number or kcat value of the corresponding catalyst. Here, the turnover number of complex 1 is 278.4 per hour and that of complex 2 is 300 per hour. Thus, in conclusion, I have synthesized 2 tetranuclear oxime-based nickel-2 complexes, and both the complexes show excellent catechol-like activity towards the aerial oxidation of 3,5-ditertiary butyl catechol to corresponding quinone. I acknowledge my PhD supervisor, and these are the reference. And finally, thank you to all for your kind attention. Thank you.
Thank you, Dr. Das, for your timely presentation, for nice presentation and timely presentation. So, quick questions, if any, 1 or 2 questions. Here is a question for you. Can you see the question?
Yes, I can determine that both the structure by X-ray crystallography. Yes.
No. Before that, there is another question. What is the importance of catecholase oxidation to orthoquinone in plant system?
Actually, catecholase oxidase is a natural enzyme. In our laboratory or many other researchers are interested to prepare some model compounds which acts or which catalyze the similar process.
Okay, and the second question was the resolution of the X-ray study.
Yes, I determined both the structure from X-ray crystallography.
And third question, did you check the magnetic interactions?
Yes, I checked the magnetic interaction of the both synthesized complexes, and they show mixed magnetic behavior, partially ferromagnetic and antiferromagnetic. And I published this paper already in inorganic chemistry.
So many questions which I think time will not permit us. But Dr. Das, I request you to answer to the questions whatever have been posted in the chat box. May I request you to answer to the questions, sir?
Yes. Yes, I do.
So many people got inspired. So now may I request
Thank you, sir.
May I now request Dr. Shiv Shankar Das from Government Degree College, Salboni. Dr. Das, are you there?
Lokhikanto, please close your presentation. Otherwise, hello, Dr. Das.
Dr. Das, can you close your presentation?
No. Dr. Das, please close your presentation. Yes. Okay.
Now, Dr. Shiv Shankar Das, are you there?
Please unmute.
Yeah,
Dr. Shiv Shankar Das, please unmute. Unmute.
Yes, sir.
Yeah.
Yes.
Please
Yes.
make your presentation. Please make your presentation.
Okay.
And I also request everyone, you just keep your presentation open so that you can simply share it. Okay, Dr. Shiv Shankar Das.
Can you see my slide?
Yes.
Can I start?
Huh.
Go to the slide. So,
Go to the
Yes.
yes.
now you start.
After all of you, today the presentation title is a renewable nano-sized triterimates continuously self-assembled into flower-like architecture and its application. As the petroleum-based chemicals are depleting very fast, the day will come when it will be completely exhausted. So current state of research is to use the more and more plant-based resources as the plant-based chemicals are renewable and as well as non-toxic to nature. So I try to extract some molecules from the nature and extract a beautiful molecule from the bark outer of the white rocks and extract the pure form, and the molecule is between. As anticipated, molecule is insoluble in water, but it can be soluble in some tunnel water, gives water, in water, and form, and self-assembly, and in some cases become the gel. This is the genesium is result of the molecule. When we go for optical microscopy images of the self-assembly petroleum, which is beautiful flower-like architectures and some fibers in oxidizing DM and metazizing solvent. And when we go for the electron microscope, the similar result is obtained the flower-like spherical objects obtained which are consisting of some petals inside. And similar result in other solvent parasiling also staying the flower-like architectures of the self-assembly bitumen in the parasite. And when we go in the orthodichlorobenzene for the first time, we saw that there is a flower consisting of petals, and petals are also consisting of the fibers. And the results of the mesitylene also showing the consisting results, the flower-like cell bitumen molecules. Then we go for the X-ray diffraction pattern of the meat powder bitumen and the zero gel. In most of the zero gel, it shows that there are the lamellar pattern of this molecule. Appears spectra of the meat powder is the zero gel. It's showing the shifting of the OH region, showing the hydrogen bonding of the molecule. And this hydrogen bonding type helps utilize the power expected for the absorption of the proteins like rhodamine and the classified. And beautifully show that the rhodamine incorporated on the outer side of the self-assembly structure of the bitumen. Then we go use for the another use of this self-assembly bitumen for removal of the toxic dyes, and it beautifully when the self-assembly bitumen are added to the dye mixed water, it can purify the mixed water. It really for removal of the dye from the water. This is the how dye can be removed from the water. And this is the dye encapsulated self-assembly bitumen, like crystal violet, rhodamine CMP, and this is the list of publication, and I acknowledge from the Vidyasagar University that I did the end of the my PhD, and thank you.
Dr. Das?
Yes, sir.
Okay, thank you very much for your nice presentation. Now the session is open for questions. Any questions? Any questions for Dr. Shiv Shankar Das? Any questions for Dr. Shiv Shankar Das? If not, then
Sir,
let us.
please call Gajendranath Maiti. He was most probably ready.
Yeah. Now I would like to. Yeah, you are absolutely right, Dr. Maiti. Now may I invite Dr. Das, could you please close your presentation, Dr. Shiv Shankar Das?
Yes.
Thank you, Dr. Das. Please mute your phone.
Yes.
And now may I invite Dr. Gajendra Maiti. Gajendra Maiti,
Yes.
are you there?
Yes, sir.
Yeah, can you make some presentation, please?
What's it?
Professor Maiti,
Yes, sir. Yes, ma'am.
at first open your PowerPoint.
হ্যাঁ পাওয়ারপয়েন্ট খুলেছি আগে থেকে
এবারে আপনি আপনার নিজের পিকের উপর ক্লিক করে বড় উইন্ডোটা খুললে তার নিচে প্রেজেন্ট নাওতে যান। আপনার নিজের পিকচারের উপর ক্লিক করুন। নিজের উইন্ডোতে আপনাকে দেখতে পাচ্ছেন?
হ্যাঁ ইয়েস ইয়েস
এবার নিচেই দেখুন প্রেজেন্ট নাও আছে ডান দিকে।
হ্যাঁ ইয়েস
ওখান থেকে এ উইন্ডো থেকে আপনার স্লাইডটা দেখা যাচ্ছে?
cannot share your screen
হ্যাঁ শেয়ার
cannot
ও
screen dismiss লেখা আছে
সামথিং প্রবলেম।
স্যার আপনি কি মিনিমাইজ করে রেখেছেন?
না
খোলাই আছে, কিছু একটা প্রবলেম হয়েছে।
ওকে,
you must grant permissions in order to screen share.
Then Professor Ponda, please carry on. Next speaker. I'm trying to contact.
Dr. Gajendra Maiti, you please send your presentation material to Dr. Tiki Maiti.
Yes,
He can
yes,
do the presentation on your behalf. You just have to say go to the next slide. Dr. Maiti, would it be possible?
Sir, I'm trying. Please send me through email.
Yeah, that's
Please,
what I'm asking.
please
send
give me
your
the email ID.
Okay. Hello.
Well, I think I should go for the next speaker.
Yes, sir.
Yeah. The next one is Dr. Dipankar Rai. He's from St. Xavier's College, Kolkata. Dr. Dipankar Rai. Yeah, take your presentation. And after Dr. Dipankar Rai, we will listen from Dr. Kumar Saha. Dr. Dipankar Rai. Dr. Dipankar Rai is from St. Xavier's College, Kolkata. Please. Dr. Dipankar Rai. Dr. Dipankar Rai, are
Hello.
you there?
Hello, Dr. Dipankar Rai.
Is Dr. Deepak Kumar Saha there? Dr. Deepak Kumar Saha.
Yes, sir.
Dr. Deepak Kumar Saha, please make your presentation.
Yes, I'm here.
Yeah, please. Please start.
Hello, sir. Hello.
Yeah. Dr. Deepak
Okay.
Kumar Saha, yes.
Okay, sir.
Dr. Saha, at first open your PowerPoint on the laptop. Not in minimize mode. Open your PowerPoint on the laptop. Yes. No, you cannot open this. Dr. Saha, hello.
Okay.
At first, you present your PowerPoint on the window first.
Okay.
You open. Then go to Google. দেখুন তলাতে যে গুগল চিহ্নটা আছে গো টু গুগল চিহ্ন। এই মিটিংটা চলে এসছে, মিটিংটা এসছে, আপনার নিজের ছবির উপর ক্লিক করুন। করেছেন? হ্যাঁ, আপনার নিজের ছবিটার উপর ক্লিক করুন। হ্যাঁ হ্যাঁ হ্যাঁ, ঠিক আছে, করুন। হয়েছে?
হ্যালো,
হ্যাঁ করেছেন?
আবার ওই সেই অ্যাপ্লিকেশন উইন্ডোটা চলে আসছে।
ও
হ্যালো অ্যাপ্লিকেশন উইন্ডো।
স্যার, প্রফেসর পন্ডা?
হ্যাঁ, বলুন।
Sir, please carry on. And Dr. Shah, please call me. Please call me in my phone.
Dr. Deepankar, are you there?
Yes, ma'am. Yes, ma'am.
Okay. Please start.
Yes, ma'am.
Yes.
Okay, let us first listen from Dr. Deepak Shah, then we listen to Dr. Deepankar Rai. Right? Dr. Rai, your presentation will be immediately after Dr. Deepak Shah. Okay, Dr. Rai. Dr. Shah.
Okay. Thank you, sir. Good afternoon, everybody. I want to present a poster just now. The topic is low-level cloud occurrence and atmospheric ozone formation over Kolkata, India. We have analyzed ozone data in the period 1992-1995 and low-level cloud occurrence data over Kolkata. Now, we have plot these cloud, low-level cloud occurrence data and ozone data, and then we draw a conclusion. Now, in the figure number 1, it represents the nature of diurnal variation of clouds for each month and their mean variations. Diurnal cloud variation of all months followed nearly the same trend with their mean variation. It is evident from the plot that the cloud occurrence gradually increased from 230 IST to and then gradually increases and attain maximum at midday, that is 1430 IST. It is also verified by the coefficient of correlation, which is very high. And now in figure number 2, this plot represents the diurnal seasonal variation of a cloud. From the plot, it is evident that the cloud occurrence gradually increases from the month of January, then attain maximum in the month of June to July, and then gradually decreases. It is also verified by the value of coefficient of correlation, which is very high, that is 0.95 to 0.99. Then we have plotted the seasonal variation of ozone concentration and their mean variation at Kolkata. From this figure, it is clear that the seasonal variation of the ozone concentration for every year and their mean variation followed nearly the same trend. The value of coefficient of correlation is very high, that is 0.94 to 0.98. The amount of ozone concentration gradually increases from the month of January at a maximum in the month of June to July. And finally, we plotted the cloud occurrence data and concentration of ozone. And the plot clearly shows that the concentration of ozone had increased in cloud occurrence in Kolkata. And now explanation. Cloud occurrence increases means water content in the cloud increases, which is dissociate to give atomic oxygen, which is finally combined with oxygen molecule to give ozone molecule. So concentration of ozone is increased. Another reason is low level cloud occurrence. There are the good reflectors of solar radiation, which plays a key role in enhancing photochemical reaction for ozone formation. Now, final conclusion. We can say that low-level, large and more cloud occurrence reflect more solar radiation, which enhance the photochemical reaction and supply more amount of water vapor, leading to the formation of upper tropospheric and lower stratospheric ozone. So we can say that low-level cloud occurrence significantly influence the formation of ozone concentration in the atmosphere, and this is the overall poster. And finally, I can say everybody, thank you. Hello. Hello.
Thank you, Dr. Saha, for your nice presentation. So,
thank you, sir. Thank you very much.
There are any questions? Any questions from the participants? Yeah, there is a question for you. Do you find any comparative data of ozone depletion with other metro cities?
No, we are working about this in the various places of the in India, like Srinagar, like Mumbai, etc. And there is a relation. There is a relation between ozone and cloud occurrence.
Okay, Dr. Saha, thank you very much for your nice presentation. Could you please close your presentation? Yeah, thank you very much. Now, may I request Dr. Deepankar Rai of St. Xavier's College, Kolkata. Dr.
Yes,
Rai?
sir.
Yeah, are you
Yes,
ready?
sir.
No, great.
Yes, sir. My presentation is visible?
No.
No, it's showing stop. You are presenting to everyone. Stop presenting and so on.
Yes.
Yeah, perfect.
Yes.
Please start.
Visible, no?
Yes, visible.
Okay.
Please start.
Good afternoon, everyone. Good afternoon, everyone. My presentation topic is identification of two cobalt-tolerant bacteria from galvanizing industrial sludge. We all know that cobalt is a naturally occurring element. Cobalt has some beneficial effect on human health. It is a part of vitamin B12, which is essential for human health. But when too much cobalt is taken into our body, the harmful effects can occur. Serious effect on lung, including asthma, pneumonia, wheezing, etc. There are some chemical approaches for metal removal, but these methods have some disadvantages. Another process, which is biological, we emphasize on that, the bioabsorption process. Bioabsorption can be defined as the ability of biological material to accumulate heavy metals from wastewater through metabolically mediated or physiochemical pathways of uptake. Algae, bacteria, fungi, and yeasts have proved to be potential metal bioabsorbents. So, in my study, sludge samples are collected from galvanizing industries in Tikiapada, located in Howrah district, West Bengal. From the sludge samples, initially 8 samples are isolated of different colony color and morphologies. In the meantime, the cobalt content is measured by acid digestion and atomic absorption spectroscopy technique and revealed that 8.89 milligram per kg or ppm. According to this result, 6 times and 12 times metal concentrations were set and tolerance is analyzed. From this study, it is revealed that S1 and S5 have the highest tolerance in 100 ppm, and these were selected and named P1 and P2 respectively. P1, both strains are motile. P1 is gram positive bacillus shaped and P2 is gram negative rod shaped. By biochemical analysis, both strains are indole negative, therefore they have no tryptophanase enzyme. They can produce various acids like formic acid, lactic acid, succinic acids, but cannot produce acetyl. Again, they can produce citrase enzymes by which they can utilize citrate as a sole source of carbon by breaking down it to oxaloacetate and acetate. As they are aerobic organisms, naturally they produce catalyze enzymes. Among the two among the P1 contains cytochrome C oxidase or indophenol oxidase, which transport electrons from donor compound to acceptors. They also use urease to break the amide bonds in carbon and nitrogen compounds with liberation of ammonia. P2 can ferment every sugar, but P1 cannot. Next, the LD50 of the two strains. Next, the LD50 of two strains are analyzed, and they are 250 ppm and 200 ppm for P1 and P2 respectively. Now, all the experiments were done in this parameter. For identification, 16S rRNA and same analysis has been done. From 16S rRNA, P1 is identified as Bacillus cereus, and P2 is Enterobacter species. But for more confirmation, fatty acid methyl ester study or FAME study has been done.
Please make the conclusion. Could you conclude?
Okay. Both strains bioabsorb cobalt in stress conditions such as pH, temperature, and biomass significantly. It is observed that both strains absorb more cobalt in high pH than low pH. Near about 76 to 82% of cobalt absorption occurs in pH 9, but 79% of cobalt absorption in pH 7. In neutral pH, Bacillus is more efficient absorber. In both pHs, both strains have growth curve in lower margins compared to the control. Both the strains are efficient cobalt absorber in 37 degrees centigrade, near about 70 to 80%. And both strains we have showed that antagonism effect. Both strains are also not antagonistic.
Dr. Are you there?
Most probably line disconnected, sir. Left the meeting, sir. No.
He's still there presenting. He's saying.
Yes, sir.
Let us wait for one more minute for him. Tithi, what do you suggest?
Yeah. Yes, sir. 1 minute for. Most probably connection problem, sir.
Yeah, I understand. Dipankar?
No, sir.
No, probably he got disconnected. Okay. Now,
yes.
could you resolve the issue of Gajendra?
No, sir. Actually, Dr. Gajendra Maiti will present tomorrow, sir.
Okay, fine. Then should I invite Dr. Amar Hens from Government Degree College, Ranibagh?
Yes, I am here, sir.
Okay, Amar Hens, you are here. Hello. So can you go to your presentation? Can you go to your presentation, please?
At first, open your PowerPoint.
Okay, I am in Android mobile.
Okay. The method is same.
Yeah, I know. Is it visible?
Or Hens is from Government General Degree College, Ranibagh.
Is the slide is visible, ma'am?
No. The slide is not visible. Yeah. Yes.
Is it visible?
Yes. Yes.
May I start?
Yes, sir.
Good afternoon, everybody present here. Good afternoon, sir, and thanks to college to organize such type of international webinar. Thank you, ma'am, to give me a platform to present my oral presentation. The topic of the oral presentation is enhanced sensing behavior of N4O3 heptadentate Gym2 complex in mixed aqua solution. Now, for this, we have synthesized such type of heptadentate ligand which is activated as H3L which react zinc perchlorate hexahydrate in the ratio of 2:1 and gives a well-yielded complex. And this is the crystalline structure of the dinuclear complex. And in the solid state crystalline structure, both the zinc are penta-coordinated, but not in equal geometrical environment, as the tau value are not the same. Now, we also performed the optimized structure in solution state. An interesting observation is shows that in solution state optimized structure, the environment is different with respect to in solid state, and the tau value in the solution state for both the Zn1 and Zn2 is different than the solid state structure. As we details observe the what type of elongations take place in solid state or the solution state, and this table clearly indicates that in the each bond length is elongated in the solid state from the solution state, and the maximum elongations take place here present as a asterisk O4 and O5. These O4 and O5, particularly the coordinated oxygen atom, reside in water molecule. That means in solid state to solution state, this water molecule is more elongated. That's why if there is present of suitable nucleophile, it may be substituted, and that's why we perform the UV-based titration of this complex in presence of different shape anion. It may be spherical like fluoride, chloride, bromide, may be tetrahedral, but surprisingly there is no change observed in major case, and the changes appear only in the sulfate anion. The lambda max of the complex, which appear 290, is shifted to bathychemic shift in the presence of sulfate anion. For the detailed investigation, we also perform the UV-based titration of the dinuclear complex in presence of sulfate stepwise, and the inset is clearly indicate that the ratio of the dinuclear complex and the sulfate anion is 1:1. That means this sulfate anion is displaced the water molecule and kept like bridging moieties. For the details investigation, we also perform the theoretical study TDDFT, and this TDDFT clearly indicates that the in the picture B, please, picture B, the lambda max appears 308 nanometer is mainly responsible for the contribution of the sulfate anion. That means the sulfate anion is bridging in the dinuclear complex by displacement of the water molecule. Now, we further details, we also perform the emissions property. First of all, I say that the dinuclear zinc complex is good emissive property and show blue emission light in presence of UV lamp. When we added different type of anion, no such change is observed, but in the presence of sulfate only, there is quenching on the emission intensity, which is picturized in figure D. And for the detailed investigation, we also perform the stepwise addition, and we see that when the ratio becomes 1:1, the plateau region is appeared. That means there a complex is formed, which is known as sulfate of bleach, and this is the histogram. Histogram clearly indicates this dinuclear complex is selective and sensitive towards sulfate anion by emission property, and we are unable to synthesize the crystalline structure of the sulfate bridging dinuclear complex, but in the solid residue of 1B, which is abbreviated the bridging sulfate complex presenting here red, clearly indicates that some interaction of the sulfate group and the zinc moiety is taking place.
Dr. Haynes, please conclude.
Okay. And lastly, we also saw the NMR mass spectra in the solution state, which clearly indicates that the complex is formed and the molecular mass of the dinuclear complex is completely disappear in figure B. And finally, with conclusion, we have to say that this two water molecule is substituted by sulfate anion, and by which the emissions property of the dinuclear complex is become blue to colorless. There is so much reported journal which is mainly favorable for the phosphate anion sensor, but sulfate is still layered. Now let's end thanks to all, and I also acknowledge my professor, my supervisor, Professor Kajan Krishnaraju, Jalpur University. Thanks you all for being present.
Thank you, Dr. Amar Haynes, for your timely and nice presentation. So questions from the participants, if any?
Yeah.
Questions? Any questions from the participants? If not, let us thank Dr. Amar Haynes. Dr. Haynes, could you stop? Could you just
Okay, sir.
stop your presentation? Yeah. May I now invite Dr. Devlina Sarkar from Bhagnan College. Dr. Sarkar, are you there?
Yes, sir, I'm there.
Please, please. Dr. Devlina Sarkar, she is from Bhagnan College. Please start your presentation.
Okay, sir. Good afternoon, everyone. Today I'm going to present my work entitled Turn on Fluorescence Chemosensor based on coumarin skeleton for selective detection of aluminium 3+. Actually, I have chosen aluminium 3+ because it is utmost importance both from biological as well as from environmental point of view. And I have mainly chosen fluorescence technique because it's quite powerful due to its simplicity, sensitivity, and also tunability. So I'm directly going to the synthesis portion where we have taken 3-acetyl 4-hydroxycoumarin and then condensed it with hydrogen in 1:1 molar ratio in methanolic medium, and then we have isolated this condensed product HL. Then further we have condensed this HL with 4N-diethylamino 2-hydroxybenzaldehyde in methanolic medium, again in 1:1 molar ratio, where we have finally isolated this product. This is the final chemosensor H2S. We have characterized it several spectroscopic techniques. Here I have provided the NMR spectra and mass spectra, but not going into the detail because of shortage of time. I'm straight away going to this sensing of aluminium 3+. Here, this is the UV-vis absorption spectra of the receptor. As we can see that in the pre-receptor, there is a sharp absorption band at around 440 nanometer, but with the gradual addition of aluminium 3+, this absorption drops and a new band arises at around 530 nanometer. This is the emission spectra. We can find that in case of the free receptor, there is hardly any emission intensity at around 612 nanometer, but with the gradual addition of aluminium 3+, there is a sharp increase in emission intensity at around 612 nanometer. Here we have done a comparative study where we have taken several other metal ions and also we have given our chemosensor H2L, but we find that there is no enhancement in this emission intensity, and this intensity emission is specific for aluminium 3+. We have done the Jobs plot analysis where we find that the maximum corresponds to at about 0.5, which indicates 1:1 complexation between aluminium 3+ and the receptor H2L. Here we have added several other metal ions to H2L, and to that very same pot we have added aluminium 3+ and again recorded the emission intensity at 612 nanometer. We found that H2L is quite efficient in detecting aluminium 3+ in presence of several other metal ions also, even when they are coexisting actually. This is the pH study. From the pH study we found that complexation actually took place between the receptor and aluminium 3+ in this pH region between 5 to 8. After 8, this emission intensity actually drops, which indicates that the complex has started to break. Actually, in case of the free receptor, which is the black one, we found that with the increase in pH, the emission intensity has dropped. We have calculated the binding constant of the receptor to be around 8.2 into 10 to the power 5 mole inverse, and the limit of detection for aluminium plus is around 10 to the power minus 8 molar range. So we have tried to explain the sensing mechanism by density functional theory. Actually, our synthesized receptor can exist in this 2 ketoenol form, this 2 and 3, and also in this dienol form. This is just a proton shift. Okay. So then we have calculated the relative energy of all these 3 forms using DFT calculation, and we found that these 3 forms, 1, 2 and 3, their energy difference is quite low, and hence this proton shift is quite possible, which is responsible for the quenching of emission intensity in the free ligand. On addition of aluminium 3+, this, yes sir?
Please conclude.
Yes. On addition of aluminium 3+, this proton shift is getting hampered. Here I have done the study in case of the free ligand inside the cell line. Inside the cell line, you can find that there is no emission when the metal ion is absent. But in presence of the metal ion, there is a brilliant red emission. And this I have done with human breast cancer tissue, where also we found this brilliant red emission. So finally, I would like to acknowledge my principal from Bagnan College, Dr. Nobindu Murmu sir, who has helped me in carrying out these biological activities, and Department of Chemistry, Jadavpur University, for providing me this facilities with this NMR and spectral facilities, several others. And thank you to all for your kind patience, and also a big thank you to the organizers.
Thank you very much indeed, Dr. Devdina Sarkar, for your nice presentation. So now the session is open for questions and answers. Questions, if any? Yeah, there is a question. What is the sensitivity level for metal ion in cell line?
Yes, sir. Actually, in cell line, you cannot quantify it. We have only seen it. Like it's not. We have not determined the amount. Inside the cell line, I haven't determined the amount.
Okay. And second question, madam, did you get the crystal?
No.
And third question is from Dr. Somo Sundar Nathi, is that energy of complex 3 is seen 0.
It's relative energy. I have taken that to be 0. It's relative energy.
Okay.
Okay.
So, Dr. Sarkar, thank you very much once again for your nice presentation. Now, may I invite Dr. Maniket Chaudhary from Mizoram University? Is Dr. Chaudhary there?
Dr. Chaudhary?
And Dr. Banerjee?
Sir, please call Sushanto Manna.
Sushanto Manna. Okay. Dr. Sushanto Kumar Manna from Bidhannagar College. Dr. Manna?
Yes, sir.
Yeah.
Can you hear me, please, sir?
We can hear you. Could you make your presentation ready? You start your presentation. Dr. Sushanto Kumar Manna is from Bidhannagar College. Dr. Manna, please.
Okay. Present. Yeah, window, no.
Yes. Sir.
Save. So now I can start my presentation?
Yes.
Sir.
Hello.
Yeah, you can use. You just make your presentation.
Yes, sir. Now it is ready. Thank you. Good afternoon, everybody, and thank you to all to giving me a nice platform to present. Now my topic is the development of new methodology towards novel infused heterocycles. In the last few decades, several types of heterocycles has been developed Mainly, I wish to focus on the N-alkylated. N-alkylated heterocycles, say it may be quinalogia, quinazolinia, quinolizinium alkaloid, in addition to polycyclic besimidazole anion. In this kind, we have a...
I am sorry to interrupt. No slide is coming.
Yes, yes.
I was thinking that it is only on my computer, but in everyone's
No, sir.
presentation is not.
No. A presentation is not. It's not coming. Not coming. Dr. Manna, please close. Please close and start from first. Start from first. Totally close.
Okay.
Open your PowerPoint first. At first, open your PowerPoint.
Yes, ma'am. Yes.
And on the main.
Then not minimize it. Not minimize it. Then click on your picture.
Okay.
Then present now. Present now.
I think first you will have to click on stop presenting in your screen. In your Google Gmail.
Yes, ma'am.
Yeah. You see there is something written stop presenting. You click that.
Stop presenting.
Best.
And now you open your PowerPoint.
Yes, sir.
Now go present.
Yes, I have opened my PowerPoint.
Then to Google.
A window. Now, yes.
Share.
So is it visible now? So is it visible? Yes, I have shared this. Is it visible?
Yes.
No.
Yes.
Now it's
okay. Thank you.
If I go to slide show.
Yes.
Dr. Manna, go to slideshow.
Okay. Yes.
Slideshow option.
I have. Yes. Now, is it visible? Developing new methodology towards novel infused heterozygote. Yes.
Sir, Professor Ponna.
Now start.
Yeah.
Sir, he can start in this form.
Yeah, sure. No problem. No problem.
Okay, continue.
Okay, ma'am. Thank you.
Okay.
Now I used to develop from Ghana College new methodology on infused heterozygotes. They are very like used to in heterozygally in addition to being activated or initiated. This yes, and these moiety are very important as they have to maintain fluorescence and water soluble. How to prepare? Now I prepare this type of heterozygote from easily available, commercially available tetravalent moiety. For tetravalent moiety, they are using bromide inspiration. After that, a cup reaction has been introduced in this portion, and again after we also in diamine moiety and cyclization occurs as polycyclic process kind of property. I have added iodide and it can be soluble water on it, and now this property we both mixing. Various kind of metal have been studied: alkali metal, alkaline earth metal, transition metals, and various metal. And after that, that Fe3 plus is peculiarly sensitive. Fe3 binary band to solution of minus smaller compound. Fluorescence going has of similar where the fluorescence on again 2% on so that the ligand and so previously obtained cross.
Yes, Diman. Don't click.
By to start cell plasma in compound cytoplasm and that is in
one second.
Yes, sir.
Dr. Manna.
Yes, sir.
Presentation has disappeared from the screen.
And your sound is totally blocked. Dr. Manna, can you keep present? Will you present in the next day again?
Yes.
Dr. Manna, will you present in the next day?
Yes.
Okay.
Yes.
Okay.
So, Dr. Kitmaiki, I think there are no more presenters for this session.
Yes, sir.
Okay, so on behalf of the organizer, I thank all the speakers for kindly maintaining their time. And every presentation was splendid, has been splendid, nice and thought-provoking. Due to some technical glitches, we could not. So someone saying, please arrange Gurpal Banerjee.
Yes.
Gurpal Banerjee, are you there?
I will speak in Gurpit Kur.
Yes, sir. Yes, sir.
Okay. Sir. One speaker is there, sir.
Yeah, no problem. Dr. Bhuvan Manna. Bhuvan, are you there?
Yes, sir.
Okay, please make your presentation.
Sir, I'll not share my screen.
You do not have anything.
You first open your PowerPoint.
Yes,
You first open your PowerPoint.
I'll open it.
Okay, not minimize it. Then go to your Google Meet meeting. Then click on your picture. Click on your picture.
Yes, ma'am.
You see your picture?
No.
Your picture.
I think Bhuvan is speaking from Amritsar, right?
Ma'am, I'll click on present now.
Yeah, present now. Then a window. Then you see your presentation.
Yes, ma'am.
Share.
Okay, ma'am.
Yeah. Then slideshow. Yes.
Is it visible, ma'am?
Yes.
Okay, ma'am. Good afternoon, everyone. I am Gurprit Kaur, pursuing PhD under the supervision of Dr. Bhuvan Banerjee. Today, I am going to present my work, and the topic is development of an efficient protocol for the -2 selective synthesis of biological active E3-aryl immunoindolin 2-on derivative using a catalytic amount of mandelic acid or itaconic acid as a catalyst in water at room temperature. Next is why we choose to synthesize E3-aryl immunoindolin 2-on derivatives. Because this scaffold have various biological properties like antidepressant, anti-smallpox, antifungal, antioxidant, antibacterial, or anti-tuberculosis. These are some of the example of biologically active
synthetic E3-phenyl immunoindolin derivatives. Next is synthesis. These are some of the example of bioactive scaffold involving E3-aryl immunoindolin as an intermediate for the synthesis of many heterocycle scaffold. Next is, as we see, particle scaffold is highly active. Therefore, these are some of the already available method for the synthesis of this scaffold. In this, as we see that these use, in previous method, they use hazardous conditions like the reflux, heating, microwave, hazardous solvent like dry toluene, methanol, ethanol, hazardous catalysts like phosphorus pentoxide, aluminum oxide, or oxocalcium, or expensive catalysts. So under these circumstances, development of green protocol, use of green solvent are some of the present day requirement. Next is, after notifying number of demerit in the previous method, we thought there are enough scope to synthesize under greener conditions. So we motivated to synthesize this scaffold by using a greener pathway. We use mandelic acid or itaconic acid as a catalyst. We use, for the first time, mandelic acid is used as a catalyst in our lab. This is the structure of mandelic acid, which is isolated from the bitter almonds. For optimization of our reaction, we conduct a series of trial reaction with isatine, aniline in the presence of 20 mole % of mandelic acid or itaconic acid with water, methanol, ethanol at room temperature. From these preliminary reaction, we concluded that 20 mole % of mandelic acid with water is the optimized condition of our reaction. This is the pictorial representation of a reaction, and this is the graphical abstract of a reaction in which E3-aryl immunoindolin was synthesized by using isatine, aniline in the presence of 20 mole % of itaconic acid or mandelic acid with water at room temperature. The reaction was completed within 3 to 4 hours. We got good yield, that is 86% to 94%. Check the generality of our reaction, we use acenaphthacunone instead of isatine. And this paper is published in Current Organic Chemistry. Benefits of our method is, we use organic catalyst, that is mandelic acid. We use mandelic acid as an organic catalyst. It is isolated from the bitter almonds. The reaction carried out at room temperature. There is no need of column chromatography purification. We isolate the product by simple filtration. We use water as a greener solvent. This is the entry table of our reaction. We use 9 different derivatives of isatine as well as aniline. This is the spectral data of our representative entry 3J, in which we use 2,6-dimethyl aniline or isatine as a reactant. Then we characterize our synthesized product by using HNMR, HRMS, CNMR, and we got the O-type diagram of our compound. Next, at the conclusion, simple and mild reaction condition, that excellent yield, operation simplicity, clean reaction profile, use of commercially available, inexpensive or environmentally benign organocatalyst, that is mandelic acid or itaconic acid. We use water as a greener solvent, or are the key advantages of newly developed mandelic acid or itaconic acid catalyze efficient synthesis of E3-aryl immunoindolin derivatives. At last, I would acknowledge my sincere gratitude to Department of Chemistry, Indus International University, for providing me laboratory facilities, Vice Chancellor, Indus International University, Dr. Bhuvan Banerjee, that is my supervisor, and I also thankful to IIT Mandi for spectral measurements. Thank you for listening.
Thank you, Dr. Kaur, for your very lucid and time-bound presentation. The session is now open for discussion.
Okay.
Questions, if any. Are there any questions? Yes.
Sir, listening to?
Yes,
sir. Yes, sir. Sir, what is the correct mechanism? Why the G isomer is more preferable? Why Z isomer is not for? What is the correct mechanism?
Dr. Banerjee.
Yes, sir. Because this isomers are in.
Why E isomer is formed, Z isomer is not formed?
Yes, this is as you see in OR-type diagram. This product is in E fashion. If two groups are with higher priorities on opposite side of the double bond.
No. This question is not actually what is the why the E selectivity, why you are not Z selectivity is the question. Why the one isomer is formed, another isomer is not formed? What is the correct mechanism? This is the organocatalytic reaction.
So can you interpret, can you give this answer, sir? I am myself Dr. Bhuvan Banerjee, the supervisor of this Gurpreet Kaur. So it may be when, can you hear me, sir?
Yeah, you
okay. Now when this aniline molecule approaches towards the astatine molecule, there is a ketone at the second 2 carbon. Okay. After this aniline. Okay. So when this aniline molecule approaches the hydrogen, there might be interaction with that particular carbonyl also. So to avoid that steric repulsion, it approaches from the behind part and it selectively forms this E form.
Is there any mechanism, organocatalytic cycle type mechanism is present? Is there any mechanism for this reaction?
No, see, it is a very common and very easily understood.
No, I know actually why diastolic selectivity arises. Is there any interpret? Only the static interaction is the factor.
Dr. Shubhankar Sarkar, I think you have the contact address and you can post your question and Dr. Bhuvan Banerjee will be happy to answer to you.
Thank you, sir.
You can discuss, you can post your question or comment in the chat box and I think Dr. Bhuvan Banerjee will be happy to answer to this. Right, Dr. Banerjee? Yes, sir. Yes, sir, definitely. Dr. Titi Maiti, are there any more to present paper for today, this session?
Most probably no, sir. Most probably no.
If not, then on behalf of the organizing committee, I convey my sincere thanks to all the speakers. Ma'am, can you say something once? So, ma'am, I have already provided you 5 abstracts, I think, for different presentations, and I think you have accepted 2. If time permits.
Yes, you can present in the next day, tomorrow.
Yeah. In tomorrow, if time permits, please allow us to present all the remaining 4 because all our I think listeners will enjoy their presentation. And few students are undergraduate students.
I will try.
Dr. Bennett, you can discuss it personally with Dr. Titi Maiti. Please discuss that with Dr. Titi Maiti. Okay. I think more than happy to answer.
Okay.
So, and last but not the least, let me convey my sincere gratitude to all the organizers.
Gurpreet Kaur, please close your presentation. Yes.
Yeah. So I convey my sincere thanks to Dr. Maiti and the organizing committee and the principal of your college for giving me the opportunity to chair the session. And I over to Dr. Titi Maiti again. Dr. Maiti,
Thank you, sir. I convey my sincere gratitude to you. Thank you for accepting my invitation. Thank you, sir. Thank you. And I want apology to all of the paper presenters due to shortage of time because some write-up is coming in the chat box. Just coming in the chat box, the time is very short. But due to the large number of paper presentation, I have to short the time. I want apology to all of you because you are all a good researcher and you have more material to present. But as an organizer, I have to control all that thing. So please forgive me for shortage of time. If you have, I provide. Thank you. And I will all meet tomorrow 10:45 because Professor Debashish Rai is the first speaker of this technical session 1 in tomorrow, and the session will be start at 11 a.m. So we will meet in the Google Meet in on 10:45 tomorrow, 10:45 a.m. And if any type of problem create in this day, but today I want apology to all of you from organizing committee. Thank you, thank you, thank you, Professor Ponda, and thanks to all the participants. And I expect all those are present today must present tomorrow, and must you all with us will be asked with us in the tomorrow. I will wait for all of you. Now, good evening, and the session is closed for today. Thanks to all, and it is a problem. I am unable to give more time to present.